Good evening and welcome to the Brian Crombie Radio Hour on Saga 960. I've got a real pleasure and honor this evening of introducing you to one of my smartest classmates from Harvard Business School, a guy by the name of Bruno Holthof, who is the Chief Executive Officer of the Oxford University Hospitals Foundation Trust, and very involved in what's going on in regards to COVID-19 and a vaccine that the Oxford hospitals are currently developing. I got to tell you, this man is pretty impressive. So I met him because we were in school together at Harvard, where he got an MBA. But previous to that, he got an MD, and also subsequent to that, or previous to that, he got a PhD. So he's a doctor in both medicine as well as a PhD, as well as an MBA. He, after school, joined McKinsey, became a partner at McKinsey, and then ended up working in a bunch of different healthcare environments. I won't go through them all, but is now Chief Executive Officer of this hospital. And it says here that your hospital employs 12,000 staff across 4 hospital sites and 44 other locations. That's massive.
That's correct, Brian. Yeah, we have a turnover of about £1 billion. So it's indeed a sizable operation in the UK.
And yet you're in such modest surroundings right now. I understand this is because you're actually not in the UK. You're in Toronto, quarantined because you want to go see your grandson, I understand.
It's the granddaughter, and she was born indeed in Toronto in December last year, before the pandemic. And we actually visited her in Toronto before the lockdown and the pandemic started. But this will be the first time that we're back, and we're following all of the rules. And I'm sitting in the flat of my son. They're in Vancouver, and I will join them after I can be released from the quarantine period. I'll join them in Vancouver Island.
Well, it's an honor to have you here in Toronto, and a real honor to have you on my show. So, Bruno, tell us about the vaccine that you're developing. What's the status of it, please?
Yeah, we're currently in what is called the phase 3 clinical trials. And that means that we're vaccinating volunteers on a large scale. In the UK, we have vaccinated 5,000 people with the vaccine and 5,000 people with another vaccine that has similar side effects, so that nobody knows who got the real vaccine. And we also have already vaccinated people in Brazil and in South Africa. So in total, more than 20,000 people have been vaccinated either with the COVID-19 vaccine or the control vaccine. So it's pretty significant in terms of number of people that already have been vaccinated.
How long will the phase 3 clinical trial continue?
Well, the duration is 1 year, but we will be allowed to look into the what we call the database, because it's a blinded database. We'll be able to look into the database, and if we pick up any efficacy signals, it may well be that the UK authorities give us a temporary license to vaccinate more people, and we'll have to see how quickly we pick up that efficacy signal.
10,000 plus people is a fairly large clinical trial, and 1 year is fairly long. Is that standard for vaccine trials?
Oh, yeah. It's absolutely standard for any development of a drug or a vaccine. So these phase 3 trials are always large scale. And especially with vaccine, you want to make sure it's absolutely safe, but also that it works. And that's why we need these large numbers of people.
You must have done preclinical work or other studies that have suggested you think that the efficacy is there already, correct?
Yeah, we have, this is a platform that was used before for other vaccines. A similar SARS vaccine was developed for the MERS epidemic, if you recall, a while ago. And so we were able to show that it was safe at that point in time, but couldn't go all the way to the efficacy trials because the MERS epidemic was contained a lot quicker than now with COVID-19. But I think what is unique in the vaccine development now is that we started all of the phases of the development that you normally go through step by step. They were all started simultaneously at the same time. So what you normally do is you have preclinical studies that's in the lab and on animals. You wait for the results. If they're positive, you go to a first human trial, which is a relatively small scale, what's called the phase 1 trial, just to show that it's safe to go into humans. And once you have those results, you go into a phase 2 trial, a bit larger, and you try to prove the efficacy. And if you've done that, you go through the large scale phase 3 trial to again demonstrate the safety and efficacy. So in this development, all of that was started simultaneously. We developed the vaccine using the genetic code of the virus that was detected in China, which was in January, mid-January. We received the genetic code. A team of scientists in Oxford then genetically engineered the adenovirus, which is the platform we're using to insert the genetic code that codifies the spike protein of the virus. And so as soon as we had a genetically modified virus, we started producing it into a manufacturing facility. We started doing lab tests with the vaccine. We injected the vaccine into animals, monkeys, which are closely related to the human race. But we already started discussions with the regulatory authorities on the phase 1, 2, and 3 trials, the design of those, and we were able to start recruitment of volunteers at the same time as we're administering the vaccine to the monkeys. So everything was done in parallel, not just the clinical trials, but also the manufacturing of the vaccine and discussions with the regulatory authorities about potential approvals of the vaccine.
We're chatting tonight with Bruno Holthof, who is the Chief Executive Officer of the Oxford University Hospital System, about the vaccine that Oxford is developing for COVID-19. We're going to take a break for some messages and come back more with Bruno in just a minute. Stay with us. Welcome back to the Brian Crombie Radio Hour, Saga 960. We're chatting tonight with a friend of mine from business school, Bruno Holthof, who is the Chief Executive Officer of the Oxford University Hospital System. 12,000 staff across 4 hospital sites and 44 other locations. Before that, he was CEO of the Antwerp Hospital Network. And during that period, he transformed it into the most profitable hospital group in Belgium, which I guess is pretty impressive. And before that, he was a partner at McKinsey & Company, one of the global management consulting companies. He served a wide range of healthcare clients across Europe and in the United States, and has gained great experience. And he's got an MBA, an MD, and a PhD. And I've argued with him on numerous occasions over cases in class. He's one of the smartest guys you'd ever want to meet. And so it's a real pleasure having him. I've actually gone out drinking with him a few times. Well, won't talk about that at all. Bruno, you mentioned a couple of things. Genetically modified virus. What's a genetically modified virus?
Basically, you modify it in two ways. One, you make sure that it cannot replicate by itself, so that it's not dangerous. It cannot sort of replicate and transmit itself, which is required for safety. But secondly, you also insert a genetic code that when you administer the virus into a human body, that body develops an immune response against the virus and against the code that you've inserted into the human body.
You talked about this protein spike. What's a protein spike?
Well, the SARS-CoV-2 virus has a core, and it has around it a number of what we call spike proteins. When you see the visual, I'm sure that your listeners have seen it, the spikes that are demonstrated when they show the image of the virus. The virus is using those spikes to enter into human cells. So it's through those spikes that they attach themselves to a human cell, and then they enter into the human cell to be able to replicate themselves, and therefore that's the way that they also cause a lot of damage in humans through that replication in human cells. So what we're trying to do is create a response against those spike proteins, and that is both a response from cells that will attack the virus, but also the so-called antibodies, which are also proteins that will bind with the spike so that it prevents the virus to entering a human cell.
And so what happens? The virus gets in your body and you just expel it?
Yeah, it basically cannot replicate itself.
So it just lives in your body and then just doesn't replicate, so it doesn't take over your systems.
Correct. Yep. Yeah.
You mentioned that you started developing this with genetic material that you got from Wuhan in January. We've had this big controversy both in Canada and the United States, I'm not sure about the UK, in regards to how quickly governments, or frankly, how not quickly governments responded. Sounds like you were aware, or the scientific community was aware of COVID-19 in January and started developing vaccines right away. And you must have done that because you came to some realization that it was going to be an issue that needed a vaccine, if not a pandemic, a big issue. Why are we, getting all this information that governments didn't sort of respond until March?
Well, I think we all knew. I recall actually reading first about it in December of last year. So we all knew there was an epidemic, not a pandemic yet, in China, because of the infectious diseases unit in Oxford that had experience with similar sort of infections in the past. I referred to the MERS epidemic. They knew that this could become a pandemic, and certainly when the infection transferred to Italy, we knew that it was going to be difficult to contain. So we started preparing for the research, but also for the response locally against what would become a pandemic. And we sort of knew that in the month of February. So to be honest, I think in January we were focused on, let's develop a vaccine. Potentially, we could use it to fight the epidemic in China. But as soon as it left the Chinese borders, we knew that was going to be very difficult to contain, and that eventually also the UK would be a victim of the pandemic. So that's, I think, why we moved so quickly. The fact that we were preparing for a pandemic also led us to prepare the shutdown of the university, and actually of many of the activities in the hospital, which freed up a lot of talent to focus on this development.
You've talked about how you were developing in parallel. You've talked about you've got 10,000 people that are in the trial, 5,000 on the vaccine, 5,000 on the placebo vaccine in the UK, and people elsewhere as well in the world. It almost appears like there's this race going on around the world for these different vaccines, and there's competition, but then is there cooperation as well? Tell us, if you could, sort of the status of international competition and cooperation in development of a vaccine.
The competition is against the virus, is not against the scientific teams. There's actually quite a lot of collaboration across the scientific teams globally. So I think it's to the benefit of the human race that we're having this competition against the virus. There's a lot of exchange. One of the companies that is also quite ahead in the vaccine development is a company called Johnson & Johnson. Their pharma company that has developed the vaccine is based in Belgium, my home country. So I know the people very well. We had lots of exchanges between the development of Janssen Pharmaceutica, a Johnson & Johnson company, and the Oxford development. So there wasn't a race against each other. It was more a collaboration, an exchange of views, and how to move this thing as quickly as we could to fight the pandemic.
But there does seem to be some competition that even different nations are almost trying to get the vaccine developed for them and buy as much vaccine supply as they possibly can. Is that a problem?
I think there's also a lot of, again, joint purchasing. If you look at the Gavi sort of alliance, which is purchasing a lot of the vaccines, which I think is a good thing because these companies need advanced purchases to be able to fund the whole development program and the scaling up of the manufacturing on a global basis. We need here billions of vaccines to vaccinate the whole of the world. So I think it's quite good that you have these purchasing coalitions being formed and making commitments on the most promising vaccines and betting on different horses, because we never know which ones will be the effective ones.
There's been a lot of press about this Moderna vaccine that's a different kind of development. Can you explain and describe that, please?
Yeah, Moderna and the BioNTech Pfizer vaccine use a similar methodology, which is different from the Johnson & Johnson and Oxford vaccine, which, as you know, is being commercialized by AstraZeneca now. So AstraZeneca, J&J use a adenovirus vaccine. Moderna and BioNTech Pfizer use a RNA vaccine, which basically injects the RNA with the same aim of developing an immune response to the spike proteins of the SARS-CoV-2 virus. The big difference between the two platforms is that for AstraZeneca and Johnson & Johnson, you just need to have vaccines that are kept cold, like in a normal fridge. The Moderna and the Pfizer vaccines require frozen vaccines, so at extremely cold temperatures to keep them stable, which is one of the differences in terms of how you're going to have to distribute and inject the vaccines going forward.
So that's how you store the vaccine. But my understanding is that the Oxford, the J&J, the AstraZeneca use a inactive part of the vaccine, of the virus. Is that correct? And the RNA is a completely different kind of process?
Yeah, Brian, there's actually three modes of actions that are now in phase 3 trial. You just referred to inactivated virus, which is the Chinese vaccines that are under development. And they're in phase 3 trials, for example, in the UAE. They use inactivated virus, which is a methodology used also with other vaccination programs. The Pfizer and the Moderna technology is using genetic code, but the RNA code of the virus to induce an immune response. And the Johnson & Johnson AstraZeneca vaccines actually use another virus, so not the COVID-19 virus, but another virus, and manipulate the genetic code of that virus with the code for the spike protein of the COVID-19 virus.
Fascinating. And you think all three might work?
I mean, so far, all three methodologies have published data in well-known scientific journals, well-respected scientific journals, and they have shown promising results. What they've shown so far is that they induce an immune response, pretty similar to what people will develop when they get the infection through a normal transmission. What we don't know is whether that immune response with people that have developed the infection or people that have been vaccinated is protective against a new infection.
And is that because the virus mutates or just because it's not effective?
No, we need to just have enough statistical data to show protection after that people have been vaccinated, and we don't have that yet for none of the vaccines.
No, I understand, but typically when you get vaccinated and you've had a virus and you've created the antibodies against it, you wouldn't be able to get the virus again. You wouldn't be able to get infected again. And you're saying you want to check that. Is your concern that the virus is mutating, or that it just can't get immune?
It is a strong hypothesis that it will work, but you have to prove the hypothesis. And I mean, there's some other viruses that after a first infection, when you get it again, actually you get a worse infection. The dengue fever is a good example of that. And that, again, has been sort of discussed in media as a possibility with any viral infection. And we just want to exclude that possibility and indeed demonstrate that if you've been vaccinated with any of these vaccines, that it protects you against the virus. And we don't have that data yet.
So you've got more than 10,000 people in your phase 3 clinical trial. Do the other horses in the race have comparable kinds of people already enrolled in the phase 3 clinical trials?
We're ahead in terms of numbers of people vaccinated, but all of these companies that we've discussed have large-scale phase 3 trials already started. It just takes a while before you come to the sort of the 10,000, 30,000. In some cases, we'll probably go up to potentially 60,000 people that we want to vaccinate.
So with all these horses and 10 to 60,000 people times 3 different horses in the phase 3 clinical trials, when do you think we'll know whether it works? When do you think we'll have an approved vaccine? When will it start being commercially produced and being sent out to the general public?
That's the $1 million question, Brian, that we get asked so often. And it's very difficult to predict because we don't know which one will pick up the signal first. Increasingly, people are confident. When we said in Oxford that it would be before the end of the year, we said it in the sort of June, July timeframe of this year. We were not taken seriously when we said that. Increasingly, people around the world are believing that we could have one of these vaccines picking up a efficacy signal before Christmas. That doesn't mean that the world will be vaccinated before Christmas, because as I was telling you, we need billions of vaccines, and the logistical effort required to then start vaccinating people is enormous. So to give you an example, we're now, of course, preparing for the vaccination program in the UK. Oxford will be the hub for vaccinating the southeast of England, but just the whole logistics of storing the vaccines, distributing the vaccines, having the manpower to at least give one dose per individual. And for some of them, we may need 2 doses of vaccines, another dose a month later. This is going to be a significant logistical effort. We hope if we get efficacy signal before the end of the year that we will start vaccinating healthcare workers. Those are now also included in our clinical trial. So quite a number of my staff members are enrolled in the phase 3 trial. It was, I can tell you, overly subscribed. So a lot of people wanted to be in the trial. So I expect a big sort of take up in healthcare workers because they're very exposed, of course, to a second wave and potentially a third wave. And then I think the other big question that I'm sure you would otherwise ask me, Brian, is who will we vaccinate first? Because you can't vaccinate everyone at once. So I think most countries will vaccinate healthcare workers first. And then it will depend on the which vaccine shows efficacy and in what we call it target groups. There is a big concern that vaccines may not work as well in older people, which, as you know, are the most vulnerable against this virus. But vaccines tend to be not working as well in older people. So again,
why is that? Because it doesn't create enough of an immune response, or what?
Immune response when you grow older becomes less strong. And so
can you just give them a bigger dose?
Well, it has to. It's about the dose, but also the frequency. So you can give them what is called a booster dose, which is a second dose later on to boost the immune system. But you need some level of protection, which again is sort of assessed through statistical methods. And so that will be the big question. Some countries that have a very young population, especially in Africa or some of the Middle East, Asian countries, are therefore also contemplating vaccinating the younger population to create the so-called herd immunity. If you protect people in the younger age groups, they can't transmit the virus, and therefore that way you protect the older population. So that is still up for debate, and we'll have to wait for the efficacy data to decide what is the best vaccination strategy in each country.
But you think that we'll actually have an efficacy signal by Christmas of 2020, and so we might be actually able to start vaccinating people early in the new year?
That's correct. Yeah, I mean, and the vaccines are available. I mean, all of these companies are producing at scale, and the sort of current planning is that hopefully 2021 will be the year of vaccination, and I mean, some of the authorities worldwide are saying hopefully by the end of 2021 we'll be in a very different place.
Did you get the vaccine?
I didn't enroll in the trial, Brian. Do you know why? Because I wanted to come to Toronto to visit my granddaughter as we started the conversation.
Well, you'll probably get vaccinated when you get back. We're chatting with Bruno Holthof, the CEO of Oxford University Healthcare System. We're going to take a break and come back more. And I'm going to ask Bruno about some of the other things that are relevant that I know Oxford is working on, things like tests. Stay with us. What a pleasure it is tonight on the Brian Crombie Radio Hour for me to be chatting with Bruno Holthof, who is the CEO of the Oxford University Healthcare Hospital System. Great guy. He's lived in Belgium and worked in Belgium at a big hospital. He's now in the UK, CEO of a big hospital system that's working on a vaccine for COVID-19. And he's actually physically today in Toronto doing the 14-day self-isolation, which is the government rules before you can be let out to go see his granddaughter who he's visiting in Canada. So it's a pleasure to have Bruno here amidst us. Hopefully we'll get a chance to grab a Belgian beer or something like that before you leave. Bruno, we were chatting the other day about a couple of other things, and you were saying that you were in a validation study for saliva tests. Can you tell us about that, please?
Yeah, I think one of the key tools that we will use to fight the pandemic is faster testing. I mean, I know you're very interested in opening up the hockey league to large crowds, Brian. And of course, when you want to open venues like that or movie theaters, you need better testing, and better testing means faster testing that allows you to have more point of care results. Because right now the gold standard is that you take a swab of your nose and throat. That cotton swab goes to a central lab where the material is extracted. It has to go through a large machine. That takes hours. Yeah? And most labs have a turnaround time of maybe up to 24 hours before you get the result back. That isn't very helpful if you want to organize a hockey game or if you want to screen people before going into a movie theater. So what we ideally like is that we have a very cheap test that can be used before people go into the pub or a movie theater. They just can take the test, prove that they're negative, and then therefore give some confidence to themselves and to their fellow colleagues and the owners of the pub or whatever, that they're not admitting people with the virus into the venue. Currently, what we call the PCR tests that are done centrally cost about $25. These saliva tests will just cost a couple of dollars. So in terms of deployment, if we can validate some of these tests, they could be deployed at scale. There are a couple of tests that have been validated around the world, and companies like Abbott are scaling up production so that they can be made available at large scale. but we're throughout the pandemic, actually starting in February, we've been organizing enormous validation exercises for all of the PCR and saliva tests in our hospital system.
So tell me, how is this different? The swab up the nose or in the throat gets sent to a central lab, and what are they looking for versus the saliva test? What is it taking? Is it genetic material like the swab, and what is it being put in to test it?
That's a good question. So in the central lab, in the PCR test, it looks at the genetic code. The saliva test actually tests against the antigens. That's the spike protein that is called an antigen, and the protein developed by the immune response against that spike protein is called the antibody. So the saliva test will pick up the antigens of the virus, so the proteins.
And it's as efficacious, you think, as the swab test?
The gold standard will still be the swab test and the PCR. Because, I mean, when you look at testing, you need to make sure that you avoid any false positives and false negatives. In the jargon, it's called the sensitivity and the specificity of the test. When you want to do some screening for large venues like the hockey game, you can probably afford to miss a few cases, because you'll probably miss them because they don't have a lot of virus in their respiratory system, so they may not be as infectious as what we call the super spreaders, which have lots of virus in their airways. And these tests are sensitive enough that they will pick up most likely the super spreaders, but they may miss some people that actually have a virus in the respiratory system. If you want to use it to really be sure that that individual doesn't have the infection, the PCR test will still be the gold standard, because they're very sensitive. And especially
is there a statistic that you use to describe this? I think you called it sensitivity and specificity, the difference between the gold standard and the saliva test?
Yeah.
Is it like 90 versus 95% or something like that?
No, we're looking for a test that you want to use to, for example, screen a patient before he or she goes into the hospital. You want to have sensitivity specificities of 99.9%, really high. If you just want to do screening to at least give some additional comfort before going into large venues, people will accept lower percentage points.
What kind of number do you think is reasonable?
I mean, 92 could be fine if you're just screening.
And the saliva test, you take it, what is it, little swab that turns red, or do you put it in a solution, or what?
Like a pregnancy test, yeah. It changes color.
Right away.
Well, you have to let it work for a while, but they're quicker. I mean, there's now so-called PCR tests that don't have, so the cotton swab doesn't have to go to the central lab. You can actually analyze the genetic material in a point-of-care device, and that could be deployed, for example, in an emergency department. When a patient comes in, you take the cotton swab, you put it in literally a cartridge. The cartridge will do take the material out of the cotton swab, will do the sequencing in the machine, and you'll get the results 19 minutes later. So that
19 minutes.
19 minutes for the PCR testing.
And when do you think these saliva tests will be validated and commercially available?
Well, some of them already are commercially available, and the validation is probably something that will, I mean, some of them have claim validated results. We've been asked by the UK government to validate some of the claims that these international companies are making.
We also talked about the antibody tests. Are you involved in any validation or investigation of antibody tests?
The antibody test, again, is we've done, for example, we screened all of our staff members for antibodies. That's again another test. It basically indicates to you whether you've had the infection. Because if you had the infection, you will have developed some antibodies against the antigen of the virus. And so we've done screening of our health care workers using antibody testing, which is again a different methodology, but not similar to the quite similar to the antigen test, but you're just testing for another protein.
And so these are widely available today as well?
They're not widely available yet. No, and they are available. And again, for example, I mean, some of the commercial companies do have antibody tests available, and we're using them at scale now.
So, I've had some personal experience here. I was at a conference in the United States where 10 people came down with the infection, and I was asked to self-isolate. I wanted to get the test, and I was told I had no symptoms and don't show up because it's, number one, you're going to put yourself at risk for getting the virus for coming to a lot of the hospitals or testing facilities. And it's going to be a week before you get the results back. And so you might as well just stay at home and make sure you don't get sick. Then I went in for surgery and I was given the swab test and they took me into the surgery before they could get the results. And so, I'm not sure what good the swab test was, but they needed to do it before they took me into the surgery. And I am convinced I've had the virus, and so I've been looking for an antibody test, but I can't seem to find one anywhere.
Well, I can't specifically talk about Canada, Brian. I would have to look into the availability of antibody tests in Canada. It would surprise me you would not have it. But I mean, the first two examples, is of course when you have capacity limits on testing, which most countries still have. There's not just enough machines and people and reagents. Most countries limit the PCR testing to individuals that have developed the symptoms. We have done asymptomatic staff testing and actually indicated that quite a number of staff were carrying the virus but had no symptoms. So they had to self-isolate because they were potentially a danger to patients and colleagues. But then we traced all of their colleagues and patients to see if they had infected any colleague or patient while they were asymptomatic but infectious. Unfortunately, we couldn't find anyone that was infected as a close contact by those asymptomatic staff members. So that gives a bit of confidence that testing symptomatic people is probably a good strategy to do, because you are more infectious when you develop symptoms, and it has to do with the viral load that you carry. And if you have a higher viral load, you're more likely to have symptoms than if you have a low viral load, no symptoms, but you still carry the virus. And if you would do a test, it would be positive.
Well, that's interesting because, I think the reason why we went through this lockdowns was because we thought that asymptomatic people were infectious enough to infect people. And so therefore, we closed down our economies. Or what you're suggesting is if asymptomatic people don't typically have enough viral load to infect people, why don't we just tell people that are sick to stay home rather than tell everyone to stay home?
Well, I think the lockdown measure was to just cut the transmission of the virus altogether. So it was not because some people would otherwise transmit the virus. It was more to avoid people getting the virus while out and about. And I think as we know more about the virus, because a lot of what we're now discussing wasn't known in March or whenever the lockdown measures were introduced. So as the society becomes smarter, I'm sure that the lockdown measures will also become smarter. And that's what most countries are now discussing.
Gosh, I hope you're right. I hope you're right in that regard. Based on the conversations and reports that I've read, it's difficult to get this virus outside, and you need prolonged exposure in a poorly ventilated space inside to get it. Is that correct?
Well, again, more evidence needs to be collected, but whatever evidence we have, indeed is that you need close contact for a certain amount of time. We don't know exactly how long, but it's not a fast sort of contact that will get you infected. We've used 15 minutes of close contact as a rule, but that's not evidence-based. And of course, when you are in the open air and well-ventilated environment, you're less likely to catch the virus because it is.
Why is that? Is it just sort of one germ doesn't do it? You need a constant flow of the germs into your.
You need a sufficient number of You need a sufficient number of viruses to be in your airways. Again, reminding you that the spike protein of each individual virus is used to enter a human cell, but you need a sufficient number of these viruses to be able to enter a large number of cells to be able to replicate themselves. And then you're able to transmit the virus, but also you potentially become ill.
Really? That's fascinating. The other thing that I've read recently is that they did a study in China and found that there were very few people that wore glasses that caught the infection, suggesting that maybe the infection actually got into the body through the eyes. Is that possible?
Yeah, there's been, and there's been also reports that ophthalmologists and people working close to the eyes or the nose were more vulnerable to the infection. So whether it's through the eyes or through the nose, we don't know exactly, but it may well be that there is a transmission. I would not say necessarily through the eyes, but closer contact probably led to increased transmissions.
Well, the implication of this Chinese study was that, frankly, maybe masks weren't the solution, but we should all be wearing goggles. Does that make any sense?
Masks definitely make sense because we know for sure it's transmitted through the airways. I don't think there's evidence that it's transmitted through the eyes.
Okay, well, that's interesting. And one of the other things that's interesting about this virus that you probably are well aware of is that people are saying it doesn't just have a respiratory impact, it has a vascular impact. Can you explain that?
Yeah, it's basically, it's an overreaction of the immune system to the viral infection. So you of course get respiratory symptoms, but because of an overreaction, you also get lesions into the vascular system, which is it's sort of called an overreaction, and it has led to some of the complications like renal failure and other complications as a consequence of the infection.
I understand that there's been things like blood clots and even blood clots that have gone into the brain, and so therefore people have got long-term cognitive impacts. Is that true?
Correct, yeah. So it's also a vascular disease, if you call it that way. Yeah.
Unbelievable. Well, this is a fascinating conversation we're having with Bruno Holthof, who's CEO of the Oxford University Hospital System. We're going to take a break and come back with a couple of final minutes in just a minute. Stay with us. Well, we've been having a fascinating conversation tonight on the Brian Crombie Radio with Bruno Holthof, who is the CEO of the Oxford University Hospital System. 10,000 people, 4 hospitals, 40 units. My gosh, it's a big operation. Bruno, what about remedies? What work are you doing from a remedy standpoint? What do you think about? I think we've heard about remdesivir. We've heard about low-level. What is it? Low-level amounts of steroids. We've heard about this one that Donald Trump was focused on. Where are you on remedies?
Yeah, besides the vaccine development and the testing, which we initiated in March, the other big initiative that was started was a large-scale clinical trial of any COVID-19 patients in the trusts, but also in the whole country. So it was a countrywide effort, and it was called the Recovery Trial. And we basically enrolled all of the COVID-19 patients admitted to hospitals in Oxford, but also countrywide into this trial, and randomized the patients to different treatment protocols. as we would normally do for a clinical trial. And we got very quickly statistical results, some of them in a positive sense, that remdesivir had some impact on improved outcomes in intensive care. But a very cheap drug that you refer to, cortisone, also had a significant improvement of outcomes in intensive care. And rather than wait for publishing that result into a scientific paper, as soon as that data was unblinded and we made that discovery, it was made public through a press conference. And worldwide, that treatment protocol was adopted almost immediately because it had such a big impact, and it was such a cheap and widely available drug. On the potentially also positive side, we proved that hydrochloroquine doesn't work. So we proved that some drugs did work and other drugs didn't work. So that was quite valuable as well, so that we could dispel some of the myths that you would have to take this anti-malaria drug and you would be better off than if you didn't. So that's not true. We're still enrolling patients into the trials, and we now have monoclonal antibodies which are being developed. These are targeted medications against the virus. So we're testing those, but fortunately, for the patients, we don't have a lot of infections right now that come to the hospital. Today, I still only have 2 patients in the hospital with COVID-19. But if there would be more patients coming into the hospital, we would again randomize them. And if we have sufficient patients on these new drugs, we could potentially show that they have a significant impact as well.
What is a challenge trial?
A challenge trial is basically, if you want to demonstrate the efficacy of a vaccine, you have to wait for the natural pandemic to infect people and to show that people that are vaccinated don't get infected, and those that didn't get the vaccine are getting the infection. But you're at the whim of the pandemic, and for a vaccine developer, they want to have lots of virus circulating in the society, which is not what we want. But if you want to show efficacy through the natural transmission, you need lots of people that get the infection through normal contact. The challenge trial basically allows you to actively challenge the vaccinated individuals with the virus and prove that they are protected against the virus, which of course is a very high-profile trial, because you have to manufacture the virus to be able to administer a dose of virus into the airways of an individual. And you would not do that from an ethical point of view if you did not have a treatment that works, because you don't want to have a person that you actively infected end up in intensive care. So we are waiting for some of the outcome of the trials we just discussed around monoclonal antibodies, for example, to see if they work even better than remdesivir or cortisone. And we call that a salvage therapy that is good enough to be used to rescue people if the vaccine would not work and you challenge them with the virus.
Bruno, a lot of skeptics are saying they're not going to take the vaccine. Either they're anti-vaxxers or they don't believe that COVID-19 is real. What do you say to those people?
Well, first of all, I will take the first efficacious vaccine, Brian, and I will do it for myself, to protect myself. But if also the data shows that I can protect my friends and family, I will do so. And I'm a very strong believer in vaccination programs. If we would do some vaccination programs around the world, we would eliminate entire diseases such as measles. Unfortunately, still too many people die of measles today, and that's because of the anti-vax movement. There's also a lot of urban myths circulating. And to give you an example, one of the first volunteers, we took a picture of her and posted that on social media. And that same picture was used by the anti-vax movement to say this person has died. So we had to convince this person to retweet, and she retweeted, "I'm very well, thank you. I'm having a cup of tea, alive and well." But that's quite damaging when people create distrust against vaccines and vaccination programs, because the only way we're going to get rid of SARS-CoV-2 is by having mass vaccination.
The only way we're going to get rid of SARS-CoV-19 is through massive vaccination.
Exactly.
Bruno Holthof, who is the CEO of the Oxford University Hospital System since October 2015. He employs 12,000 staff across 4 hospital sites and 44 other locations, who is developing one of the 3 horses in the race to develop a vaccine, is probably going to be one of the people that changes our life for the positive in 2021. And thank you, Bruno, for the work that you and all of your staff and researchers and scientists and doctors are doing. And thank you so much for coming on our show and explaining it to us tonight.
A pleasure, Brian.
I look forward to having a beer with you before you leave Canada.
Yeah,
thanks very much. That's our show for tonight. Thanks for joining us. Good night.